Hello again, listeners. Welcome to the podcast of the InnCoCells project. And this edition of the podcast comes from Nancy in the northeast of France, where we're currently enjoying the 7th InnCoCells project meeting, particularly all the great presentations which have brought us right up to date with the recent progress from the project. So in this edition, I'm happy to welcome Rebekka Leitner from InCoLogic. And Rebekka, welcome to the podcast. It would be great if you could start us off by introducing yourself and tell us a bit about InCoLogic and what it does.
Okay, so hi, as already said, my name is Rebekka Leitner and I'm working for InCoLogic Technologies based in Vienna, Austria.
So what does InCoLogic Technologies do on a day-to-day basis?
InCoLogic Technologies believes in the potential and value of microorganisms to our planet and our everyday lives.
Is this your mission statement?
Exactly, because so we are therefore working on the formulation and of the production of microorganisms, which can be used as a key to unlock various of ways and possibilities to contain sustainability. And we are a very young team of scientists and engineers from over 10 different nations, I think.
Oh, right, okay.
Yes.
How many people work there?
Now 35.
Okay.
So we grew quite a lot.
Yeah. And how long has the company been operating?
Since 2016.
Right, okay. So it is a very young company.
Yeah.
And it's in its early growth spurt. Interesting.
Yes, totally. I worked there as an expert engineer for now 5 years. I'm responsible in our organization for scale-up, which can be, for example, fermentations, meaning cultivations in bioreactors, as well as formulations like drying processes. Additionally, I focus in my daily work on data evaluation, which is for me the backbone for upcoming decisions, process development, and optimization approaches.
Right, so essentially what you're saying there, if I understand correctly, is not only do you do the experiments with the cultures, but you also evaluate the data and work out what the cultures are doing and maybe use that information to feed back into the next set of experiments.
Exactly.
Wow, that's really interesting.
Yes.
Okay, well, I've been looking forward to exploring that in detail with you. However, before we do that, quite interested, I mean, obviously a very specialised topic. So how did you get there? What was your career route to get to work on scaling up cell cultures and data analysis in that context?
So InCoLogic Technologies is also kind of working, has a mission, which is also related to where I come from. So I grew up in a very, very small village next to the Hungarian border.
Okay.
In a small farm, so agriculture and plants and nature and sustainability was always something where my heart goes.
So it's been like a dream mission to get here.
Exactly.
Amazing. Okay. Yeah.
So I started to study bioprocess engineering, so bioengineering and food and biotechnology. So biotechnology was in my daily life since I'm 15.
Oh, wow.
Yes.
What were you doing in biotechnology at 15?
There is a school, an agricultural school, so like farmer school, which has a special education for food and biotechnology.
Right, okay then. So you were already interested in it at this early age. And I mean, I think most of our listeners will know what biotechnology is, but maybe bioengineering is a slightly more specialised category. Could you maybe elaborate on that a little bit, what that might involve?
Okay, bioengineering is more focusing on the equipment and on how you set up this equipment and use the equipment for your goal. Exactly. So it's more technical, not only studying in labs a microorganism and how the genetics changes. This is more another branch, but bioengineering is more, okay, we produce now a ton of microorganisms, but how can we do this?
A few weeks, maybe months ago now, I did a podcast with Jaya from LIST and possibly explored a bit of a similar topic about how to make microorganisms behave, in this case plant cells, but similar concept, how to make them behave how you want at different scales. So it sounds like there might be some similar themes there. Anyway, sorry, I'm interrupting you because you were going to tell me about some chain of events from your very early biotechnology career up to where you are now. So take us away.
Yeah.
How did you get from being interested in bioengineering at age 15 to working at InCoLogic?
I did my master thesis at InCoLogic, and there I already started to work with the hairy roots. So hairy roots is a plant tissue And we focus on the cultivation of those hairy roots. They look like hairy roots, obviously, the name saying. And they are naturally occurring. So there is a bacterium, it is called agribacterium, which transforms those tissues in like this three-dimensional root-like structure. And we can use those roots, for example, for the cosmetic industry to produce secondary metabolites or also peptides. They find also usage in the pharma. So they have a potential. And what I did in my master thesis is to study how we can produce those hairy roots in bioreactors.
Right, okay.
And there I started with very small bioreactors.
So I'm starting to see the link now. So your interest in bioengineering intersects now with this idea of taking what's generally a normal plant tissue, which grows into the soil, but you can infect it with this bacterium, agrobacterium, and suddenly the roots are transformed into a constantly growing tissue with lots of extra hairs, which is why they're called hairy roots. And these, if the natural roots produce the metabolites, then the hairy roots are like an endless source of the same molecule. Brilliant. So this almost pre-answers my next question, which is how you got involved with the InnCoCells project. So presumably this is all tied up with the mission of the InnCoCells project to produce metabolites which are used for as cosmetics. So I can see the link, but how did you hear about the project? What was your pathway to joining? Have you worked with any of the partners before, for instance?
Unfortunately not. But the story is basically my boss came to me and was like, hey, you are our expert for hairy roots. I think you're the perfect person for the InnCoCells project. So let's do this and make it work to scale the hairy root production up.
Right, okay. I know that there are a couple of other partners working on hairy roots. So do you take their cultures and scale up, or do you start the whole process off yourself too?
So we have partners like VTT. They send us their hairy roots in petri dishes, like very small amounts. And what we did then in-house is to first create an own cell bank, like a backup cell bank, and then start from the petri dishes into tissue culture flask, from there into bioreactors, and then in even bigger bioreactors.
I see.
And also do like optimization studies. So how can we increase the yield, and how can we make sure that the conditions that we have in the bioreactor are perfect for the growth or also for a target molecule production.
So it's not like a case of taking a sample you're given and throwing it into a massive vat. You've got to kind of do it in steps. And they get used to growing in a slightly bigger system and then a slightly bigger still. I'm presuming there must be some sort of unique aspects of this process. You can't just take them and put them into another bigger dish and they grow. There must be some kind of process for making it work properly. Maybe you could take us through some of those things.
It depends also always on the species or on the hairy root line. So generally speaking, you have to differentiate a little bit between the hairy root species and lines. But in general...
Why is that? I mean, what causes the difference? If you'd think, somebody who's a novice, why is there a difference between this species and this species?
They also look a little bit different. So the genome is different, so all the pathways are different. So there is no general answer to this.
Is it like, because I'm not an expert in this, my background is developmental biology, so I understand it now, a mouse develops differently from a chicken.
Almost, close enough.
There are lots of similarities, and I suppose between two plant species which make roots, there are also similarities, but the differences are enough that you have to pander to those differences, if you like, to make them grow exactly how you want to in a particular dish.
For example, you can use different media, or some grow better in light, some grow better in the dark. So it's like this simple that there are already differences. Is it dark or light?
This is going to sound like a really stupid question, but why do roots need light?
So that is one of the benefits of the hairy roots, that they don't need light in general. I just said that they behave a little bit different when there is light.
I see,
So the hairy roots themselves, they don't need photosynthesis, but they have sugars, so they use sugars as carbon source. So I hope that this answers your question.
No, so not being an expert, it's very interesting for me to hear the description that's too hyper-technical, because for our listeners, as I said, we're in the project meeting, And what we've been listening to is some very highly technical presentations, lots of very detailed data, completely impenetrable, I would think, for someone who doesn't work in the field. So it's very nice to get this sort of like top-down view from someone who really knows what they're talking about. So no, that's great. What I would like to ask you then is, because you do this every day, there must be some specific problems you encounter when you're doing this work. What would some of those be, and how do you get around them?
So one of the biggest challenges for the production of hairy roots are for me that they grow in this three-dimensional structure. So for example, imagine you have your plant at home and you remove the soil, and then you have like these clumps of roots, and in the middle of those clumps sometimes there is not much air or water, or simplified said, not so much exchange of nutrients. So for the hairy roots, it's quite a challenge to achieve a very good condition all over the structure of the roots. So in the middle,
uniform access to nutrients there. Yeah, okay.
Exactly. So here are some challenges, and also how to, let's say, get them out and harvest them, because they form this very tight and strong structure. You have to really rip them apart.
Oh, wow, okay.
Yeah, it's very strong. It's like
Velcro.
Yeah,
almost, yeah.
Exactly. Yeah. So what we also developed while the InnCoCells project is a fragmentation process, for example, inside a bioreactor to fragment and also to harvest the hairy roots.
So they don't need to be contiguous then, like they don't need to be all attached. They can be separate parts and they still grow.
Yeah.
Oh, okay. That surprised me, actually. I somehow seem to imagine you'd need a system, because they're roots. There'd need to be
Yeah. So, but when you inoculate the bioreactor, you have many parts, like a lot of fragments.
So they're more like cells then growing than actual organs. They are self-contained and they behave autonomously in terms of processing nutrients. They're not like an organism as such.
You mean one organism?
Yeah.
It's a hard question. They are an organism.
Because I had this idea that it was like a root system, and it would be one thing. Whereas a cell culture, the cells are separate things.
Okay, now I get your point. Okay, so basically you have many root systems in one reactor.
Right. So they basically
not only one big one.
So they're not sort of like dependent on one single. They can be autonomous, smaller sections. Right.
Yes.
Okay. Good. Okay, that's it. So do you get any problems if you? I don't know how many we're talking about. Obviously, I guess there'd be different numbers of individual root parts in different scales, but do they ever sort of like go wild and start behaving differently from what you're expecting?
Yeah. That's a very interesting thing. When you forget, for example, to have the hairy root flask just on the table, and there comes light and whatever, sometimes they form even small leaves, like they get green.
No.
Yes,
Really?
Yes, and it looks amazing. It looks really weird, kind of alien, kind of. Sometimes they just change completely.
So how would you describe that technically, that kind of, where one part of the system changes independently of the other?
So they change in morphology, you mean?
In morphology?
Yes.
Okay, so it's like their structure and shape changes.
Exactly, and also the color, which is also fun.
So you can easily see them in the context. And what do you do? Just pluck them out and throw them away? Or
normally I'm stop working with them.
Yeah.
Because the genome, so the metabolism is not the same anymore. So they change due to by accident, for example, or by having them in the light. So something changes, but then I will just stop with working with them, and we always have a backup in the cell bank. So if you're working with hairy roots, you need always a backup.
Yeah, you mentioned this right at the beginning. So the first time you receive a sample, you set some aside, and then if everything goes really badly wrong, you've always got another.
Exactly.
So you mentioned the fact that you stop working with these roots that misbehave, let's say. Changing morphology. And when that happens, so what you're talking about is basically a process of selection. You pick out the stuff you want and you weed out the parts which are misbehaving. So you end up with maybe not a homogeneous collection of roots, but something that's doing more or less what you want. And you mentioned that you're particularly concerned about the metabolism changing. And this, I guess, is because ultimately, certainly in terms of this project, you're looking for these roots to produce something.
Exactly.
Yeah.
So those roots, they can produce bioactive molecules, for example, secondary metabolites, which can be used in the cosmetic industry, but also peptides, for example, also interesting for the pharma. Right. So bioactive molecules, you can target them, you mark them, for example.
You mark them?
Yeah, we also talked about it today in the presentations. And then you specifically, let's say, pinpoint those roots who produce those target molecules and then continue working with them.
So you take them out and you put them into a new dish and
for
you
example,
carry on.
Exactly.
I see. So,
exactly.
So basically you're taking the ones which do exactly what you want and then you're purifying the selection and whatever grows from that selection should also do what you want. So you were saying that as well as the metabolites, which can be very useful for the cosmetics industry, you can also do bioactive peptides, which are, if you like, more pharmaceutical than cosmetic. So do you, or have you, worked in other Horizon projects with other partners doing that kind of thing, or was InnCoCells your first Horizon project?
Oh, InnCoCells is my first one.
Oh, congratulations.
Thank you. But I really enjoy it.
Yeah.
So
it's a great
maybe
atmosphere.
Next one.
It's a great atmosphere, though, isn't it?
Yes.
To work in a big consortium like this. Yeah.
Yes.
To remind our readers, we started off with 17 partners, then we dropped to 16 because one of the partners was taken over, and now we're back to 17 again. And we've just sort of had our first meeting with our brand new set of 17 partners, and it's been going extremely well. It's these are very, very friendly meetings and everyone gets on very well indeed. A lot of us know each other going back many years. Obviously, Rebekka, you're a new person into the group, but you can see how great it is to have this big, it's like a big family.
Yeah, that's true. That's true. Yeah. But I'm in the InnCoCells project since the beginning.
Yeah.
So, but it's really nice to see all the partners on an annual basis. Yeah. And it's really also interesting to get input from them, ideas, exchange new methods. So I really like it and enjoy it, and it's also quite a fun.
Yeah, no, that's exactly what it is. So, I mean, obviously these meetings are great fun because we have a good laugh together, but the serious side of it is you get to exchange data and you get to exchange advice and stuff like this, and that is a good part of the Horizon system.
Yes. Which
brings me on actually to a very similar topic. So the benefits of being in a big consortium. How do you see this having a sort of an impact on Evologics as a company in the short term or the long term? Is it something that's going to maybe not change the direction of the company, but has it had a positive impact on your work and things like that? Yeah.
Of course. You get new ideas and input from the partners who work on sometimes the same topics or similar topics, and you can learn from each other, which is amazing. For example, in process development, what do they use? What do we use?
Yeah.
How can we together find a solution for a problem, for example?
Yeah.
So that's amazing.
You mentioned VTT earlier, VTT being the coordinator of the project, but also very big in terms of hairy root cultures. So it must be quite good to work with them and share that information with them,
yes.
etc.
And they share hairy root cultures with us.
Yeah.
And we share our product with Ilvo, for example. So there is quite some exchange in knowledge and also product in form of hairy roots.
Yeah, that's good. So I've got to be a little bit careful talking about this next bit, because obviously some of the stuff we're doing in the project is confidential, but within the bounds of that confidentiality, if I said to you, how's it going? Is it in terms of how the work is going and how successful have you been in terms of reaching the aims you set out at the beginning and what you've achieved? How is the work progressing for Evologics in your opinion?
So the results are always communicated within the project and also with the partners. So there is not really a secret. So I can talk about it. So the work for the InnCoCells project, it is amazing how far we came, to be honest, and I'm very proud to say this, because we started with very small Petri dishes and now we are in industrial scale. So
which means?
In over 1,000 liter bioreactors.
Over 1,000 litres?
Yes.
Oh, that is amazing. Obviously, I know what 1,000 litres looks like, but I can't imagine 1,000 litres of hairy heavy roots. I can't imagine quite what that looks like. It must be a fairly astounding sight.
Yes. And this is a great opportunity to share also these results and also communicate our hair root technique with others. I see here a lot of potential also for further projects, further exchanges of techniques. And
Well, that was fascinating, Rebekka. Thank you ever so much for joining us in the podcast. It's been a really interesting edition. I do like the ones where we get a little bit of technical background and some sort of like discussion of how things work, what challenges there are, how those are overcome. I find that endlessly fascinating. Thanks very much for coming.
Thank you.
Thank you very much, listeners. And I'm going to be recording the next session very, very shortly. So keep tuned and we'll see you again next time.